The next group of viruses we need to discuss it is Arbovirus. And actually, if you take a look at taxonomic classification, you will not find that group because it is a collective term which refers to arthropod-borne viruses, arboviruses. Actually, that group combines a lot of different pathogens which are transmitted through blood-sucking insects. And we have different type of insects, we have different viruses included into the group, and main families are Flaviviridae. We will discuss them today. We have Tobaviridae, and if you remember that measles again, it is an exception because actually it is a family of viruses which are transmitted through blood-sucking insects. And in general, that group of viruses, arboviruses, includes more than 250 different species of viruses which are distributed worldwide, and as minimum 80 of them can cause different diseases in human. And of course, not all arboviruses causing damage of neural organs, neural tissue, but today we will discuss few pathogens which are really causing disease of central neural system. So one of the most known diseases and one of the most popular disease in tropic areas, it is the yellow fever virus. So it is a classic representative of Flaviviridae family, and it belongs to Flavivirus genus and yellow fever virus. And that disease is actually acute hemorrhagic disease. It's not disease of central neural system. But viruses are transmitted through arthropods, and they have a classic structure and classic properties of arboviral infections. So different types and different genuses of mosquitoes can transmit the disease, and usually main symptoms are jaundice and fever. That's why yellow fever. Again, jaundice is yellowish color of skin and mucous membranes. Besides that, disease can manifest with severe headache, fever, nausea, and fatigue. So initially, disease is endemic in Africa and Central South America countries with hot tropic climate. But in India as well, you have those regions, and some decades ago, and maybe even some few centuries ago, in 17th till 19th centuries, the disease was transported by tourists and migrants to North America and some European countries. So accidental cases of disease are still registered, and of course your country is suitable and one of the most suitable for that virus country, so you should be familiar with the disease as well. So virus by structure is related to West Nile virus, St. Louis encephalitis, and Japanese encephalitis viruses, and by common properties they are very similar. So classic structure of flaviviruses, it is almost common for any virus in that genus. It is icosahedral symmetry. Capsid is made of C protein. Usually it is a single protein with multiple subunits in that capsid. And it is enveloped virus surrounded by a lipid, or by a layer or two layers of lipid tissue originated from host cells membranes. So virus, of course, have peplomeres, so glycoproteins, and their enveloped antigens or enveloped proteins, and membrane or matrix M protein. So the major area of viral surface is made of E protein or glycoprotein spikes, and actually it is the most active part of the virus. In the left part of the slide, in the left picture, you can see electron photo of virus in lower mummification, and actually virus itself and precise structure is shown in the right picture of that slide. So disease is usually identified in primates, in high primates, in monkeys in natural conditions, and human again is acting as accidental host. Incubation period can last till 1 week, and usually disease will manifest with sudden onset of fever and chills, severe headache, and general discomfort, body pain, muscle pain, nausea, vomiting, weakness, and common symptom of respiratory or some other viral infections. So diseases caused by arboviruses are usually mild and self-limited, only in some low percent of cases, some clinical manifestations will appear. So usually convalescence is spontaneous and complete. In some subjects, it was noticed that common malaise and weakness can last for several months, but in general status, good and prognosis is favorable. So about 15% of people who were symptomatic after short period of remission means then symptoms are gone and people think that they are healthy and recovered. So other episodes of disease, but in more severe state, in more severe duration can appear. And usually it will manifest with high fever and jaundice. It is possible to have different types of bleeding and shock and multiple organ failure if it is internal bleeding in some organs. In that case, in the case of recurrent disease, mortality rate can reach 60% in different countries in different conditions, but usually it's not lower than 30%. And of course, it is high mortality rate for that disease. And actually, it is high mortality rate for infectious disease. And common problem with yellow fever and for multiple viral diseases, we have only symptomatic therapy. And of course, we don't have any options to release viral load. We can only decrease symptoms. To diagnose the disease, we have two types of tests. One is targeted to identify antibodies, immunoglobulins class M or neutralizing antibodies in patient serum or in patient blood. And we have ELISA and the unique method microsphere-based immunoassay to diagnose the disease in a correct manner. The main preventive measure is control of vectors, mean control of mosquitoes, wearing appropriate clothes when you are attending some forest area or park area, use of different aerosols and repellents to avoid bites of mosquitoes, common measures to avoid insects in home, protection of windows and other. And in countries where the disease is endemic, we have vaccines to prevent disease in humans, to provide specific immunity in people who are living here. So nowadays we have mainly live vaccines which contain a live complete full virus, and they are available as injections. Again, usually they are available in countries where this is endemic or in different tourist companies who are affording that trip in those countries. Second virus is more important, much more important. It is virus of Japanese encephalitis. It is the same flavivirus, and common structure of virus is almost identical to previously described virus. So one of the differences, virus is transmitted by another genus of mosquitoes. It is Culex mosquitoes, and again, usually hot countries, it is Southeast Asia or Western Pacific countries are endemic for the disease. And actually, initially it was, of course, Japan where disease was identified and was highly prevalent. But in nowadays, because of good vaccination campaign in Japan, the disease is almost absent and name is just historic. But actually, current year and some previous years, the disease is highly registered in India included, and in some countries in Pacific region. So we will talk later about in your country again that vaccine against Japanese encephalitis or JE vaccine is integrated into the national schedule of vaccination. So in major number of cases, symptoms will be very mild, not noticeable, or even absent, and only less than half of the percent people will notice some symptoms, and usually they are limited to central nervous system and manifest as encephalitis. So So in the case of encephalitis, mortality rate is up to 30%. In most severe cases, usually it is lower, but disease can be complicated with permanent disorders or sequelae in psychiatry state or different problems with central neural system. Again, similar to other diseases, we don't have any specific treatment. So naturally, different type of animals, such as usually pigs and birds, wild birds who are living near or in the water, on the water, they're acting as a natural reservoir of infection or source of infection, and human-to-human transmission is rare. Incubation period is up to 2 weeks, and again, a lot of diseases will be very mild or even without symptoms. People will notice some slight fever, maybe headache, common malaise, and other symptoms which are not specific. And in kids, or especially in younger kids, disease can manifest with intestinal symptoms such as vomiting or pain in abdominal area. And usually disease will be misdiagnosed or diagnosis will be not correct. It will be some GIT infection, but not Japanese encephalitis. In the case of severe duration of the disease, usually fever will have very acute onset. It is high fever with severe headache, rigidity of neck muscles, and different disorders of central nervous system, such as disorientation, seizures, paralytic disorders, and people can fall into the coma, and lethal outcomes again in 30% of cases is possible. In main number of cases, convalescence will be spontaneous, and again, usually about 20 or 30% of people who had severe duration of initial disease, they will have some permanent damage of neural functions. It can be different type of paralytic disorders, shaken or Parkinsonian syndrome, different intellectual disorders, and seizure syndrome, which can be recurrent or rare, but in a few cases it was few articles described such disorders, inability to speak, and up to again 60% of cases can have fatal outcome. Usually it is in endemic areas, of course in people with low immunity and who was not vaccinated previously. Diagnosis is almost the same. Control is again the same. Usually it is a fight with mosquitoes and prevention of mosquito bites. Again, we have vaccines in our country. That vaccine is available only for tourists. In some countries, it is introduced in international scale of vaccination. So, few words about that plague reduction neutralizing tests. It is a method which is performed in plastic wells, as you can see on the picture, and you need to take sample of blood from subject, obtain serum, and the main idea is to identify antibodies that can neutralize virus and prevent growth or variants in those wells. Wells are filled in with culture cell, means that cells of some mammalian cells are introduced in those wells, and if you can see that initially culture is blue, and if virus is growing, it's observed as a plague of transparent area, some like a white dots. It means that virus is growing here. So we can take serial samples of blood, usually is minimum 2, but up to 3 or as many as required, and add on that culture infected with the virus. If titer of antibody is high, then virus will not grow in that well at all, or only few plagues will be identified. If titer is increasing, then less amount of plagues will be identified. If titer is decreasing, then more amount of virus will grow on that well. So by comparing different results for different samples, we can know if titer is rising or decreasing. If it is rising, it means that subject is really ill and immune response is in progress. If titer is decreasing, then subject was ill recently, but now it is in recovery stage. And if titer is stable or absent at all, means that subject is ill with some other disease. So test is not very popular. It is more expensive than regular tests and available not in every lab, but in specialized laboratories where, especially in countries where disease is endemic, the test is available and it is one of the methods of gold standard of diagnosis. So World Health Organization approved 4 main types of vaccines for Japanese encephalitis. In different countries, we have different indications in different time periods for introduction of that vaccine. But in general, the most safe vaccine is approved for kids from 2 months of age, and upper limit is not present, means that it can be given for any adult person. And usually 2 doses are required to provide good and strong immunity. Interval between doses should be as minimum a month or 28 days. If we will talk about India, then in your country, mainly it is Genvac vaccine, Japanese encephalitis. And it is a type of inactivated vaccine growing in varicella line. And of course, that vaccine is more safe, and in general vaccine is good and immunity is strong. So if you take a look at schedule of vaccination for Indian citizens, you will identify 2 doses as recommended by WHO. One is given in a year and second is given in 2 years with some intervals available for shifting. So in common, that vaccination is appropriate and according to WHO requirements, and in your country, the vaccine is obligate for every citizen of your country. So one more disease, and it will be the last disease in current lecture, it is tick-borne encephalitis. It's not so popular in India, but that disease is hyper-endemic in Russia and different territories of Russia. So the same flavivirus, and actually it's described in 3 subtypes. It is European, Siberian, and Far Eastern. And Far Eastern was actually, again, formerly known as a Russian virus, but nowadays renamed it. But again, it is endemic in Russia. Disease is usually transmitted by bite of ticks. It is hard-bodied ixoid ticks, as again we talked recently. It is a black or brownish colored ticks with dense chitin coverage, and that virus is closely related to one virus. It is endemic in India, and some symptoms and common properties are very similar. So one of the properties of that virus, it can circulate in tick population. It can be transmitted from adult tick to eggs, from eggs to larvae, and again to adult tick. So it don't even require any host. It can circulate in tick community without intermediate hosts. But naturally, small rodents are the main reservoir of infection, but again, hard-bodied ticks can act as reservoir as well. But more correct to say that it is a vector for transmission. Incubation period is up to a month, and again, disease manifests with similar symptoms: very high fever, common symptoms of viral infection, and different type of mental disorders or damage of central nervous system, such as meningitis, encephalitis, myelitis, or radiculitis. In general, main number of diseases are favorable and have good outcomes, but it is possible to have different paralytic disorders or even lethal outcomes in the case of the disease. Usually mortality rate is low. It is officially about 1%, but in Russia rate is much higher, but official data is not available and it's not reported to WHO. The same measures of prophylaxis, control of vectors of ticks, and vaccination. So one more thing, if you will identify the tick, it's much better to take it with you to laboratory to identify any disease which is transmitted through tick bites in laboratories. Don't kill it, don't burn, put it in some vial and take it with you. We have vaccine. It is wholesale inactivated vaccine to control the disease. And actually, that's all for current lecture. Bye-bye.